STB 1093, Microbiology Group 3, Subgroup 4. Practical 1A, Microscopy Observing Microorganisms. Microorganisms are usually very small in size, which cannot be seen by naked eyes. The solution to overcome this problem is by observing using a light microscope. It can view various types of microorganisms and their structure, observe different microorganisms, and learn how to describe their morphologies. Microscope became one of the most vital tools used by microbiologists. They are able to learn a great deal about microbial cells, including their structure and morphologies. A microscope consists of two magnifying lenses, which is ocular and objective lenses. A light microscope have three objective lenses, which is low power, high dry, and oil immersion. In summary, this microscope helps students to explore the microbial world. Skills in handling the light microscope should be emphasized to use it correctly and achieve the experiment objective. A compound light microscope consists of ocular lenses, body tube, arm, objective lenses, stage, condenser, diaphragm, illuminator, coarse focusing knob, base, and fine focusing knob. Practical 1B, Microorganisms from Environment. Microorganisms are ubiquitously distributed in the environment. For instance, in laboratory, microorganisms can be found on the surface of the table, clothes, instrument, and air. Frequently, the existence of these microorganisms can contaminate the pure culture. In the microbiology research, a culture should be pure. The microorganisms are affected by a suitable environment, including temperature, pH, gas conditions, and osmotic pressure are necessary for the growth and reproduction of bacteria. Microorganisms are known for their ability to adjust themselves to changes in the environment. Practical 2. Culturing and Handling of Microorganisms. To transfer a pure culture from one vessel to another, the aseptic technique is necessary. By doing this, the microbiologist requires taking all care to prevent contamination of the culture and the spread of bacteria into the environment. Many petri plates and tissue culture plates used for growing pure cultures of microorganisms are constructed of plastic and arrive pre-sterilized from the manufacturer. Filling these vessels with a sterile media requires the use of aseptic technique. The aseptic transfer approach also shields the microbiologist from contamination with the culture, which should be viewed as a possible pathogen. Aseptic technique involves avoiding any contact between the pure culture, sterile medium and sterile surfaces of the growth vessel with contaminating microorganisms. To accomplish aseptic technique, the work area is cleansed with a disinfectant to reduce the numbers of potential contaminants. The transfer instruments are sterilized. For example, the transfer loop is sterilized by heating with a Bunsen burner before and after transferring. The work is accomplished quickly and efficiently to minimize the time of exposure during which contamination of the culture or laboratory worker can occur.
Alright, so microscopy 1A. Here are the materials. So first thing first, dust cover was removed and microscope is switched on, and then specimen slide is placed to view the specimen through the microscope, and then light source is turned on and adjusted. Specimen slide is adjusted using the stage knob, x-axis and y-axis, so that we can view the specimen through the microscope.
Eager.
And then focus knob and fine focus knob are used to make the view of the specimen clearer so that it can be viewed in HD, clearer image. So with that all done, specimen slide can now be viewed through the ocular lens. So wrapping everything up, specimen slide is removed and cleaned from immersion oil with the lens tissue, lens paper, and all of the objective lens that includes 10 magnification, 40 magnification, and further on. And finally, here are the results and interpretation for the experiment. Off and dust cover will be applied. So moving on, here are the materials for experiment 1B, microorganisms from environment. So first thing first, we did the table surface. Soap is dipped into the distilled water with aseptic technique. So not too wet, just nice. So the soap will be soap on the table surface for 3 times. Aseptically open slightly the Petri plate, then we'll brush the soap on an agar surface. Petri plate was labeled and sealed with parafilm. Petri plate incubated at room temperature for 24 hours, 24 to 48 hours. So moving on now to air. Labeling is done to differentiate between both agar plate. NA will be exposed to air for 30 minutes and PDA will be exposed for 15 minutes. So here the Petri plate cap will be open. And moving on to fingers. So first of all, finger before cleaning process is carefully pressed first. And then cleaning process with soap. Then, clean finger is carefully pressed on one segment of the other agar. Petri plate is then sealed with parafilm and incubated. Moving on to the next experiment, which is culturing and handling of microorganisms. Here are the materials. So, first of all, streaking on Petri plate. So, Bunsen burner is lit with fire and make sure the fire is blue color. And then inoculating loop is heated with fire and then rested for 30 seconds. Aseptically, single colony of E. coli is streaked. So first streaking. And then inoculating loop is then reheated and removed from fire for 30 seconds. So second streaking is done, second quadrant. So inoculating loop is then reheated again and removed from fire for 30 seconds to do the third streaking. So here is the third streaking. And then inoculating loop is then reheated again and removed from fire for 30 seconds. Here are the fourth streaking, the final streak, final quadrant. And then to wrap everything up, the inoculating loop is then reheated and removed from fire for 30 seconds. Petri plate is sealed with parafilm. Moving on to streaking on slanted agar. Inoculating loop is heated, so is the cap of the bottle to remove contamination. And then single colony of E. coli is taken, then transferred to the slanted agar by streaking, of course. And then inoculating loop is, and the cap is heated again to remove any contamination. And then labeling process is done and incubated. Moving on, culturing using nutrient broth. So here inoculating loop is heated and the cap of the nutrient broth bottle. Aseptically, single colony of E. coli is taken then transferred into the nutrient broth. The cap of the nutrient broth bottle and inoculating loop is then heated to remove any contamination. So that is done. So moving on to the other experiments, tabbing on agar, heating of inoculating wire is done first. So not that in this experiment we are using inoculating wire and not inoculating loop. And then followed by the heating cap of the agar bottle. So aseptically, a single colony of E. coli is taken, then stamped into the agar of the bottle. In the bottle. Followed by the heating of the inoculating wire and the cap of the agar bottle to remove any contamination, so that we can get the result precisely. Finally, the agar bottle is labeled. So here followed, all of the Petri plate and agar bottle are then incubated.
When it's heat, there's condensation.
Experiment 1B, Microorganisms from Environment. The first part is swab the table surface and brush it on neutron agar plates. Results: the agar surface that is brushed by the swab are grown with microbial colonies. Discussion: there are microbes such as bacteria sticking on the table surfaces. The structures that help the bacterium to attach solid surface are flagella, pili, and fimbriae. Microbes from the table surface are transferred from the swab to the agar plate and they continue to replicate by binary fission. The second part is expose neutron agar plate and potato dextrose agar plate to the air. Results: there are more microbes which are circular and irregular grown on neutron agar plate. Potato dextrose agar are grown with less microbes which are circular. Discussion: there are airborne microorganisms grow on neutron agar consists of pectin, beef extract and agar, and potato dextrose agar which made up of potato infusion and dextrose. Both are medium that supports the growth of various types of bacteria and fungi by supplying many necessary nutrients such as carbohydrate and nitrogen. However, the PDA may be supplemented with acids or antibiotics to inhibit the growth of bacteria. The third part is pressing clean and unclean thumb on neutron agar plates. Results: neutron agar plate before cleaning the finger are grown with more microbes which are circular and punctiform. Neutron agar plate after cleaning finger are grown with less circular microbes. Discussion: our skin interact with the surrounding, especially when we touch things with our hands. Thus, before cleaning our hand, there are microbes sticking on the surface of our finger. Washing hands with soap while scrubbing our hands are able to physically destroy and remove germs from our skin.
Next, I will move on to the results for experiment 2. For the experiment on culturing of microorganisms, the microorganisms is successfully transferred to neutron agar plate, and the agar surfaces that were inoculated by the inoculating loop were grown with microbial colonies. The colonies were well separated, and there are 4 colonies can be observed. For the slant agar tube, microbes can be observed as it grows according to the traces that was traced by us earlier on, and the slant morphology is filiform. For step agar, the morphological features of the step agar is filiform too. And for the nutrient broth, the sediments were accumulated at the bottom of the bottle. Interpretations: the loop is flamed to sterilize before placing into the culture to ensure that contaminating bacterial spores are destroyed. After transferring the cultures, the loop needs to be flamed before placing it on the bench top to ensure that any liquid culture on the loop will run down into the flame. If we remove the lid of a Petri plate containing nutrient agar and place the lid on the bench top, the nutrient agar will be contaminated with bacteria from the environment.
Discussion. In this experiment, we use aseptic techniques to culture and handle microorganisms such as E. coli. Aseptic technique is a method that involves target-specific practices and procedures under suitably controlled conditions to reduce the contamination from microbes. This technique can inhibit additional bacteria from affecting experiment results. In this experiment, four types of results were obtained based on E. coli strip plate agar, slant agar, step agar, and nutrient-proof medium. The bacterial growth pattern was recorded using morphological reading and recording techniques. In addition, the inoculating loop was sterilized by flaming it to prevent contamination occurs. When we use inoculating loop to obtain a single colony of E. coli from the culture booth, E. coli was transferred to the agar surface when we inoculated it. To acquire a pure culture of one species of bacteria, we separated the microbes on the agar surface with quadrant streaking to dilute the bacteria. E. coli then continues to replicate by binary fission. It grew according to the straps as how we inoculated it on the agar surface. Next, for slant agar and step agar, we can observe that the morphological trait is filiform, in which this shows that the microbes grew uniformly in an optimum temperature of 37 degrees Celsius after 24 hours. Before checking the nutrient proof, we observed a constant turbidity forming, and after checking, we observed sediments accumulating at the bottom of the bottle. The presence of turbidity resulting from microbial multiplication in the nutrient growth is indicative of bacterial growth.